inform you, look round, right? So if you were to see this slide and you see that, you might say, oh, that's a roundworm, but it's platyhelminthes. Don't look at the mouth. The mouth on all of them are round. Look at the tail end. That will tell you if it's flat, because you can tell that that's a flat worm and it's not round the whole way. And so don't focus on the mouths when you're trying to figure out which group it's in. All the mouths are round. So I've got a slide that shows you the basic life cycle. Before you get freaked out by this, you don't have to memorize all the names that are on here. Okay? You just need to know the basics of how the worm is picked up by humans and passed. So, kind of like Giardia, we're talking about a place where we don't have good, clean running water. There's not water piped into your house and then a sewage system that flushes away the waste. You're going fishing into the bathroom and doing your laundry all in the same local lake, river, that kind of scenario. So we got a guy up here fishing in the local water. This is the form of the life cycle of the schistosoma worm that burrows through the skin. Again, you don't have to know or be able to spell its name, right? But there's a form of the life cycle that burrows through the skin of the human and goes to the liver, the intestines, and the bladder, where it undergoes sexual reproduction. What gets released from the human through the eggs and feces, through the urine and feces, are eggs. So little eggs get released from urine and feces of human or other mammal that's infected. And because the waste isn't washed away to a septic system, right, or some treatment facility, it runs back down into the same local water and develops into this form of the life cycle, which goes and lives in snail. There are 3 species of freshwater snail, and again, you don't have to remember that name or any of theirs, okay, but it's a different form of the life cycle that burrows into snails than burrowed into people. And while it's in the snail, it has to be in there like an incubator. The snail is the little incubator for this worm. It will develop into the form that can burrow into humans. So if all we had was this cannot infect humans, right? It's got to develop and grow and mature in the snail into the form that can then infect humans again. So this is a necessary part of their life cycle. If the snails aren't there, they can't develop and infect us. So the alternate host is different than the reservoir host for beaver fever with Giardia, right? Beavers weren't necessary. You could go people, water, people, and reinfect us. We can't go people, water, people for this one. It's not going to happen. They have to have the alternate host to complete their life cycle and be able to reinfect people. Does that make sense? So that's all you need to know is the basics of the life cycle and what an alternate host is compared to a reservoir host. All right, any questions on schistosoma? All right. Phylum Platyhelminthes, still the flatworms. We're in the second group now, the cestodes. These are the tapeworms, cestoda. Very characteristic body style here in this picture for the tapeworms. They all have a nice round head and then a series of repeating units. The head usually has 2 eye spots. They don't see. They may possibly be able to detect light and dark. They might be able to tell it's lighter over here and darker over there, but they're not seeing shapes or color or anything like that. Okay, so very limited function on those eye spots. And then most of them have little barbs around their mouth, which would be at the top of the head. So that head region is called the scolex. It's where the eye spots and the barbs are. And so you can clearly see the eye spots and the barbs on this one. So that whole thing's the scolex. This one's smaller, but that whole round structure is the scolex. And then all of these repeating segments are called proglottids. Each one of those proglottids, each one of those boxes, is monoecious. So each box has both male and female reproductive parts in it, each segment does. And they're intended to break off from this tapeworm. Tapeworm primarily lives in the intestines of the infected organism. The scolex attaches to the lining of the intestines these proglottids are supposed to break off and leave with the feces. So we're talking about animals like pigs or cows that live in a field. One cow has a tapeworm. Some of the proglottids break off, leave with the feces, land on the grass. The next cow comes along and eats the grass nearby, takes in the proglottids. Each one of these proglottids could attach and form its own scolex to make a whole new tapeworm. And after you've had one for a while, and they grow, they get really, really long. Right? They coil themselves up in the intestines so that if you could lay them all out flat, they'd be taller than you were, because they coil themselves up in the intestines. In Victorian times, this was a weight loss plan. You'd go get yourself a tapeworm on purpose, because how they feed is from absorbing food from your intestines. So no matter what you eat, they absorb the food, so you can't. Right? I wouldn't recommend this as a weight loss technique, but it certainly works, because they're stealing your food before you can use it. There are lots of rumors still hanging around about tapeworms. I remember my grandpa, this was before I had any degrees in biology to correct him. I don't think I would have corrected my grandpa anyway. But saying that you could hold an egg or something at the opening of the anus and the tapeworm would come out to get it, catch it. That doesn't happen. You'll still find that kind of thinking out there in the population. It doesn't work that way. You have to take dewormers to get rid of these things. Humans usually, of course, we're not eating the grass that the cow pooped on, right? So we're not getting them the way that cows would get them naturally in their habitat. Occasionally, instead of going into the intestines, the worm will burrow into muscle, which of course we do eat, cow meat. So that if humans are getting tapeworms, it's usually from undercooked meat where the worm has migrated into the muscle instead of in the intestines. And I should say it's very rare for it to be in beef. Much more likely that you're going to get worms from pork than from beef, which is why you can eat medium rare steak. You're not eating medium rare pork. If you watch the Food Network, some of the chefs will cook pork to medium, go a little bit pink. I can't do it. I love pork. I will continue to eat it even though the worms are in there. But I want it all the way done. I want dead worms when I eat it. That's just my personal opinion. Okay with tapeworms? They're pretty easy to identify. They've got a very characteristic body. All right. So, second phylum now, second major category of worms, phylum Nematoda. These are the nice, smooth, round worms. Okay, so if you were to cut them and look at the cut you made, their body is round. And some examples, hookworm, dog heartworms, and pinworm. I threw dog heartworms in there because I know it's one that you've heard of. But your lab book doesn't mention it, so you don't really have to know anything about its life cycle or how it works. However, your lab book does explain quite a bit about hookworm and pinworm. And hopefully you've been reading those first few pages of your lab book for each lab anyway, but just to encourage that you do. I'm not going to say much about the life cycles of hookworm and pinworm. I want you to read your lab book and get the information about their life cycles from that. Basic symptoms. What does a person with hookworm have? What are their symptoms? A person with pinworm. Okay, so basic symptoms. How it's transmitted. How does a human get hookworm and pinworm? So how it's transmitted, basic symptoms. Where is it common? What part of the world do you find them in? And if your book mentions any treatments. I don't expect you to go to the internet and do any research. Okay, just the stuff that's in your lab book is fine. Symptoms, how it's transmitted, where is it most common in the world, and if your book mentions any treatments, those as well. All right, so what we're not going to do for this lab is go through every single slide and have you tell them apart. And that's because for this lab only, lab 5, I'm not going to make you identify each slide by name. Okay, so if you flip over to that materials page, lab 5, it's on page 41. When you look at these slides, you're not looking at them to identify them by name. What I want is for you to be able to identify the phylum that worm belongs to and which group it's in. Okay? Trematoda, Cestoda, or it's in phylum Nematoda. So you don't have to, only for lab 5, you don't have to identify them by their specific scientific name. You need to identify them by their group. So I'm going to tell you what group they're in. You're all on page 41. The first 5 slides, all of the schistosomas through Clonorchus. Page 41 we're looking at here. The first 5 slides listed under organism. Those are all phylum Platyhelminthes, group Trematoda.
You said
The first 5, all 4 schistosomas in Clonorchus. The first 5 under organism. Phylum Platyhelminthes, group Trematoda. Platyhelminthes, group Trematoda. So if you were to see a picture of one, that's what I want you to say. Not schistosoma or Clonorchus, but phylum Platyhelminthes, group Trematoda. The next 4, all 3 Taenias, all the way through Diphyllobothrium, big D word. Those are all phylum Platyhelminthes as well, but they're group Cestoda. Phylum Platyhelminthes, group Cestoda. And that was all 3 Taenias through Diphyllobothrium. Phylum Platyhelminthes, group Cestoda. What number? The next 4. All 3 Taenias through the D word, Diphyllobothrium. The last 4 Enterobias through Trichinella are phylum Nematoda. And there aren't any groups under this. It's just straight up phylum Nematoda. So the last 4, Enterobias through Trichinella. Phylum Nematoda. Those are your roundworms. So, when you're looking at a worm, you're going to know it's a worm, right? They're the biggest organisms that we look at, so you're not going to confuse them with the fungi or with the protists, right? You're going to know it's a worm. How you tell which group it belongs to. First thing I do is ask myself, is it a tapeworm? Because this is a very distinctive, easy to recognize body. If it's a tapeworm, it's phylum Platyhelminthes, group Cestoda. If I see a scolix and proglottids. If it's not a tapeworm, I don't see a scolix and proglottids, then I've got 2 options left, right? It's either a trematode or a nematode. Then I'm looking for the overall shape of the body. Flukes are flattened toward the tail. Roundworms are going to look round through their whole body, even at the tail end. And usually, not always, but usually the nematodes are brown in color. Brown is round. So I rule out the tapeworms first. If it's not a tapeworm, I look at the body shape, particularly toward the tail. Flattened, it's Trematoda, round, Nematoda, and in general, the round ones are brown.
What's Trematoda?
Flat's Trematoda.
brown is Nemo.
Brown is round, Nematoda. They're not all brown, but most of them are. One last thing to warn you about, the Trichinella slides, the last 2 on your list. Trichinella lives in muscle. This is really the worm in pork that we're worried about while we can't eat medium rare pork. So what you're going to see on that slide are a bunch of muscle fibers running across the slide. And then the worm itself is going to be coiled up in the muscle fibers, like that. Just so you know what you're looking at. But again, you're never going to have to look at it and tell me it's Trichinella. You're going to have to tell me that it's phylum Nematoda. I'm just giving you fair warning that that one's a little bit different than the other slides because it's in muscle. Question?
How do you identify Cestoda?
How do you identify the Cestoda?
Yeah.
Scolix and proglottids. They have this body shape. That's the first one I look for. Does it have a head and repeating proglottid segments?
Okay.
Any other questions? I'd like to pass on. You guys can go ahead and get started.
So we're not
Next time
identifying the
we
name,
do
we're
have
doing
that big
group and
quiz,
phylum.
then you'll have the rest of the lab just to look over the slides. There isn't any lecture next time. Where's the slide?